Welcome to SciSense, where we unwrap scientific research, because science should not be a puzzle.
Okay, Mike. I need to tell you about something that just completely blew my mind. I'm talking about molecules that scientists have been trying to make for 50 years, and finally, someone cracked the code.
50 years? That's older than the internet. What were they trying to make that was so impossible?
Right? So this paper is called Hetero 3.1.1 propellanes. I know, try saying that 5 times fast. And it comes from Rebecca Reevey and Edward Anderson's team at the University of Oxford, published in Nature Chemistry. And Mike, these aren't just any molecules. These are imagine if someone just figured out how to make a brand new type of Lego brick that could revolutionize how we build things.
Okay, but what does that actually mean for someone like me? Why should I care about these what's-its?
So here's the thing. You know how when you're designing new medicines, scientists are always looking for better building blocks, right? Imagine you're an architect, but instead of boring old rectangles, you suddenly get access to these incredibly strong, rigid, three-dimensional shapes that can fit into spaces nothing else can. That's what we're talking about here. These molecules could literally change how we make drugs.
Wait, hold on. What exactly is a propellant? Break that down for me.
Oh man, okay. So picture this. You know those old-timey airplane propellers with the wooden blades? A propellane molecule is kind of shaped like that, but in 3 dimensions. It's got this super rigid, cage-like structure that's under incredible strain, like a coiled spring just waiting to snap open. The 3.1.1 part? That's just chemist speak for how the atoms are arranged. Think of it like an address for the molecule's architecture.
And these Oxford folks were the first to figure out how to make the hetero version?
Exactly. So for decades, chemists could make the all-carbon versions. Imagine building with just plain wooden blocks. But what Reevey and Anderson figured out was how to swap in oxygen, nitrogen, and sulfur atoms. That's what hetero means. Different atoms. It's like suddenly being able to build with blocks that have magnets, or Velcro, or springs built right in.
Okay, so why was this so hard to do? What was the problem they were trying to solve?
Great question.
So the issue is that when you try to make medicines, you often start with these flat, pancake-like molecules, benzene rings, stuff like that. But here's the problem. Our bodies are 3-dimensional, right? So these flat molecules often don't fit properly into the biological locks they're supposed to open. It's like trying to open a complex 3D puzzle box with a credit card.
Right, that makes sense. So these propelling things are more 3D.
Exactly, and here's where it gets really cool. The team realized that if you could make these heteropropellanes, you could use them as starting materials to build what they call bicyclo 3.1.1 heptanes. I know, more tongue twisters. But think of these as perfectly rigid, 3-dimensional scaffolds that can perfectly mimic the shape of flat benzene rings, but with way better properties for drugs.
Better how?
Well, for starters, they're more soluble in water, they're more stable in the body, and they can reach places that flat molecules just can't get to. It's like the difference between trying to thread a flat ribbon through a complex 3D maze versus using a perfectly shaped 3D key.
So how did they actually make these impossible molecules?
This is where it gets really clever. They started with something called 2,3-dibromopropene. Think of it as a simple starting material, like flour for baking. Then they used a rhodium-catalyzed. That's rhodium, one of the most expensive metals on Earth. But here's the amazing part. They only needed 0.05% of it. That's like using a single grain of salt to season an entire pizza.
Wait, 0.05%? That's basically nothing.
Right? And they could do this on a 20-gram scale. That's like making enough material to supply a small pharmaceutical company. The rhodium helps stitch the molecules together in this incredibly precise cyclopropanation reaction. Imagine using molecular scale tweezers to fold origami, but with individual atoms.
Okay, but then what? They can't just stop with the starting material, right?
No, this is where it gets really ingenious. So they take this cyclopropanation product, and then they have to build the heterocyclic ring. That's where the oxygen, nitrogen, or sulfur goes. For the oxygen version, they use something called DIBAL-H. That's diisobutyl aluminum hydride, basically a molecular reducing agent. Think of it as chemical scissors that can cut very specific bonds.
And this actually works? They can make these things that stumped chemists for 50 years?
Not only does it work, Mike, but get this. They can store these molecules for months. Remember, these are incredibly strained spring-loaded molecular cages. The carbocyclic versions, the all-carbon ones, are so reactive they basically explode if you look at them wrong. But these hetero versions, they found that the nitrogen one with a tosyl group, that's like a molecular safety cap, was so stable they could actually grow crystals and take X-ray pictures of it.
No way! So they could literally see the structure?
They could see the structure. And here's what blew me away. The bond angles, the distances between atoms. It's like finally being able to take a high-resolution photo of something you'd only imagined. The X-ray structure showed bond lengths of 1.56 angstroms and 1.48 angstroms. That's smaller than the width of an atom, but it tells you exactly how these molecules are twisted and strained.
Okay, so they can make them and they're stable. But what can you actually do with them?
Oh man, this is where it gets exciting for drug discovery. They showed that these propellanes can undergo what they call radical ring-opening reactions. Picture that coiled spring we talked about earlier suddenly snapping open, but in a completely controlled way. They demonstrated over 30 different reactions. That's like having 30 different ways to open your molecular toolbox.
30 different ways. Give me some examples that I can actually wrap my head around.
Sure. So they could attach things like acetyl groups. That's like sticking a molecular flag on the structure. They could add benzyl radicals. Imagine gluing on molecular handles. They even did something called halosulfonylation, basically decorating the molecule with sulfur and halogen atoms. It's like having a 3D molecular Christmas tree that you can decorate with exactly the ornaments you want, exactly where you want them.
And this actually leads to better drugs?
Well, here's the proof. They made an analog of sonidegib, which is an actual anti-cancer drug. The original drug has some problems with solubility and how the body processes it. But their hetero version, the one with the oxygen atom replacing a carbon, showed improved solubility and better physicochemical properties. It's like taking a sports car and making it more fuel efficient without losing any of the performance.
That's actually incredible. So what does this mean for someone like my mom who might need new medication someday?
That's the beautiful thing, Mike. This work basically just handed medicinal chemists a whole new box of molecular Lego blocks. Instead of being stuck with flat, limited shapes, drug designers now have access to rigid, three-dimensional scaffolds that can be decorated in dozens of different ways. We're talking about potentially better cancer drugs, more effective pain medications, antibiotics that can reach previously inaccessible targets.
And they can make enough of this stuff to actually matter?
Absolutely. They demonstrated multigram synthesis. That means they can make enough material for serious drug development programs. Plus, remember that rhodium-catalyzed trick? At 0.05% catalyst loading, even using one of the world's most expensive metals becomes economically viable. It's like figuring out how to make a Ferrari using only a few dollars worth of premium parts.
This sounds like it could be huge for the pharmaceutical industry.
It really could be, Mike. Think about it. For 50 years, these molecules were just theoretical curiosities. Now they're practical synthetic targets that can be made on scale and stored on the shelf. The team even showed that some of their ring-opening products can be converted back into the original propellanes. It's like having a completely reversible molecular transformation. That level of control is unprecedented.
So what's next? Where does this research go from here?
Well, I think we're looking at the beginning of a new era in drug design. The team has shown that these heteropropellanes are just as versatile as their famous carbocyclic cousins, but with the added benefit of heteroatoms that can fine-tune biological activity. I wouldn't be surprised if in 5 years we see the first drugs on the market that started from these exact molecules.
Amazing. Well, thanks for walking me through this molecular breakthrough, Sarah. It's incredible how 50 years of impossible chemistry just became routine.
Thanks for joining us, and remember, the next time you hear about a new drug with improved properties, it might just have started life as one of these impossible propellanes.
Thanks for listening to SciSense. Share it with a friend. See you next time.